Bismillahirrahmanirrahim. Today, the topic which we are going to discuss is the resting membrane potential. And the resting membrane potential is the electrical potential difference across the cell membrane when the cell is at rest. This potential difference lies across the membranes of almost all the cells. We are not talking about the excitable cells like neuron or muscle. This resting membrane potential lies across almost all the cells, and you can see here that there are more positive charges in the extracellular fluid and more negative charges in the intracellular fluid. Another peculiar point which needs to be noted is that here you see in this slide that there is an unequal distribution of ions in the extracellular fluid and the intracellular fluid. There is slight excess of positive charges in the extracellular fluid and the slight excess of negative charges in the intracellular fluid. तो एक्स्ट्रा सेलुलर फ्लूइड के अंदर पॉजिटिव चार्जेस ज़्यादा होते हैं और इंट्रा सेलुलर फ्लूइड के अंदर नेगेटिव चार्जेस ज़्यादा होते हैं. अगर हम वोल्ट मीटर से मेजर करें, if we place a positive electrode within the cell and indifferent electrode in the extracellular fluid, the negative potential is recorded on the voltmeter. A negative potential is recorded on the voltmeter. This is a diagram representing a cell which is present in the extracellular fluid. You can see if a cross section is taken, a small part of a cell membrane is studied. We can see that the negative charges and the positive charges are attracted towards each other across the cell membrane. The negative charges and positive charges are attracted across each other in the cell membrane. The inside of the cell may be neutral itself. Similarly, the extracellular fluid as a whole may be neutral, but the membrane is polarized because the positive and negative charges are attracted along the surface of the membrane. Hence, the cell membranes are electrically polarized. More the cell membranes are polarized, or more the number of positive and negative electrodes along the cell membrane, greater will be the polarization. For example, in figure A, this is less polarized, while C, it is more polarized due to alignment of positive and negative charges along the cell membrane due to attractive influence of the positive and negative charges. More the number of charges separated, more will be the electrical potential difference. The separated charges along the membrane serve as a miniature battery, and this miniature battery can serve for the transport of the ions across the cell membrane and also for the conduction of the action potential. Now, the resting membrane potential, the values of the resting membrane potential. The resting membrane potential in large myelinated nerve fiber is studied to be minus 90 millivolt, while the resting membrane potential in neuronal cell body is minus 65 millivolt. Similarly, the resting membrane potential in skeletal muscle is minus 90 to minus 80 to minus 90 millivolt, and in the smooth muscle the resting membrane potential is minus 50 to minus 60 millivolt. In rest of the cell, this resting membrane potential is generally between minus 70 to minus 90 millivolt. In the clinical importance, this surface current or the changes in the current between extracellular fluid and intracellular fluid can be used in recording of the electrocardiography ECG, which gives about electrical conduction studies in the heart, and also the electromyography and the nerve conduction studies. as well as there is implication of this resting membrane potential in local anesthesia. Now, this is one cell which is shown in this diagram. In this cell, you can see that there is much higher concentration of positive ions in the extracellular fluid, and there is much higher concentration of negative ions within the cell in the intracellular fluid. In this, A represents the anions. The anions are the negative ions like sulfate, phosphate, and other proteins which are present within the cell and cannot cross this lipid barrier easily, cannot cross this lipid barrier easily. Moreover, there are sodium and potassium ion, which are the most important ions which play their role in maintaining the resting membrane potential. The concentration of potassium is much higher within the cell, while the concentration of this potassium is much lower outside the cell. Conversely, the concentration of sodium is much lower within the cell, and its concentration is much higher outside the cell. The concentration of sodium inside the cell is 14 milliequivalent per liter, and concentration of sodium outside the cell is 140 milliequivalent per liter. In studying the resting membrane potential, we mainly focus on sodium potassium ATPase pump, sodium channels, and the potassium channel. These sodium and potassium are the leaky channels. First of all, we discuss about the sodium potassium pump. We have discussed the sodium potassium pump previously. The sodium potassium pump has a characteristic that it can bind with three sodium ions on the inside surface of the channel, while it can bind with two potassium ion on the outside surface of the channel on the extracellular at the extracellular fluid space. In at that side on the outside of the cell, it can bind with the two potassium ion, and there is breakage of ATP by acetylcholine by the enzyme, and the ATP is broken down into ADP and the phosphate, and due to this broken down energy, this energy causes the uphill movement of three sodium ion to outside of the cells, and up and the uphill movement of the potassium ion inside the cell, because in the cell the concentration of potassium is already higher, while the concentration of potassium sodium is already higher outside the cell. So this pumps moves the ions against the concentration gradient, or it causes the uphill movement of the ions, and it will generate the negativity within the cell, because more positive ions are pumped out and less positive ions move inside the cell. So in collectively there will be negatively developing within the cell, and it develops an electrical negativity of about -4 millivolt within the intracellular fluid. Then there are the leaky channels, which are very important. We have discussed voltage gated channel, ligand gated channel. There are also leaky channel which are present within the cell membrane. And the leaky channel simply cause are the simply mediated by the simple diffusion, that whenever the concentration of the ion is higher, it will cause the greater movement of the substance across the membrane. So the sodium concentration is higher outside the cell, and hence through the leaky channel the sodium ion will move inside the cell, while the concentration of potassium ion is higher inside the cell, and through the potassium leaky channel potassium ion will move from within the cell to the extracellular fluid. However, here one thing which is very important to know is the permeability factor. the permeability of potassium ion is much higher as compared to the permeability of the sodium ion. This leaky channel, the potassium leaky channel is much more permeable to potassium ion than the sodium leaky channel to the sodium ion. So the cell membrane is about 100 times more permeable to potassium than to the sodium ion. So we will see that the sodium ion, the concentration of sodium ion is higher in the extracellular fluid. So from the extracellular fluid, the sodium ion will move along the concentration gradient inside the cell in the intracellular fluid. As more and more sodium ions are moving inside the cell, their potential or their electrical potential is increasing inside the cell, and their electrical potential would be decreasing outside the cell. क्योंकि पॉजिटिविटी जो है वो क्या हो रही है कि बाहर से पॉजिटिव आयन जब अंदर आ रहे हैं तो बाहर पॉजिटिविटी कम हो रही है और अंदर पॉजिटिविटी क्या हो रही है, बढ़ रही है। तो क्या होगा कि जब अंदर पॉजिटिविटी बढ़ रही होगी तो ये मजीद पॉजिटिव आयंस को आने से रोकेगी, और यहाँ जब पॉजिटिविटी कम हो रही है तो ये क्या है कि ये प्रोमोट करेगी कि जो है वो अंदर से सोडियम आयन जो है वो बाहर मूव करें अलोंग द इलेक्ट्रिकल ग्रेडियंट। क्योंकि अंदर पॉजिटिविटी जो है वो बढ़ रही है तो बाहर जो है वो नेगेटिविटी बढ़ रही है कंपैरेटिवली। तो इलेक्ट्रिकल ग्रेडियंट ये होगा कि सोडियम आयन विल बी मूविंग फ्रॉम इनसाइड द सेल टू द आउटसाइड। और इसी तरह इन केस ऑफ पोटेशियम आयन, पोटेशियम की कंसंट्रेशन हमने पढ़ा कि इट इज मच हायर विद इन द इंट्रासेलुलर फ्लूइड। तो पोटेशियम मूव करता है इंट्रासेलुलर फ्लूइड से एक्स्ट्रा सेलुलर फ्लूइड में अलोंग द कंसंट्रेशन ग्रेडियंट। और ये भी हमने पढ़ा कि पोटेशियम की जो मूवमेंट है या पोटेशियम की परमेबिलिटी है, इट इज़ मच हायर एज कंपेयर टू द परमेबिलिटी ऑफ सोडियम आयन। अब जब पोटेशियम आयन क्या है कि यहाँ से मूव करेगा फ्रॉम इनसाइड द सेल टू द आउटसाइड सेल, नॉर्मली पोटेशियम जो है यहाँ पे एनाइंस जो है, प्रोटीन्स और एनाइंस जो है उनके साथ बाउंड फॉर्म में प्रेजेंट होता है। तो जब इन यहाँ से सेपरेट हो जाएगा और लीक आउट करेगा इनटू द एक्स्ट्रा सेलुलर फ्लूइड तो यहाँ पे नेगेटिविटी बढ़ेगी। एनाइंस जो हैं वो उनकी कंसंट्रेशन जो है वो नेगेटिव आयन की कंसंट्रेशन बढ़ जाएगी। अब नेगेटिव आयन की कंसंट्रेशन जब बढ़ जाएगी तो इट विल पुल द positive potassium ion from the extracellular fluid into the intracellular fluid. It will pull the potassium from the extracellular fluid into the intracellular fluid, and this gradient, that is the movement of potassium ion from the extracellular fluid into the intercellular fluid due to the electrical forces, is called as electrical gradient। ये है कि केमिकल ड्राइविंग फोर्स जो है वो क्या है कि यहाँ पे हायर कंसंट्रेशन होती है पोटेशियम आयन की, और वो केमिकली जो है वो मूव करेगी अलोंग द ड्यू टू द डिफ्यूजन प्रिंसिपल्स पोटेशियम आयंस को फ्रॉम द इंट्रासेलुलर फ्लूइड टू द एक्स्ट्रा सेलुलर फ्लूइड। अब एक्स्ट्रा सेलुलर फ्लूइड में जब पोटेशियम आयन कंसंट्रेशन इंक्रीज करती जाती है और विद इन द सेल पोटेशियम के लूज होने की वजह से नेगेटिव आयन की कंसंट्रेशन इंक्रीज करती है, तो देयर इज अ डेवलपमेंट ऑफ electrical force, there is development of electrical force और या इलेक्ट्रोमोटिव फोर्स। तो वो जो है वो क्या करेगी कि वो पोटेशियम को मूव करवाएगी फ्रॉम द एक्स्ट्रा सेलुलर फ्लूइड इनटू द इंट्रासेलुलर fluid. So there are two forces acting. One is the diffusion force or the chemical force which causes the movement of potassium ion from the cell, from potassium from the intracellular fluid into the extracellular fluid. Second, due to the increased concentration of potassium ion, there is another electrical force which is moving the potassium ion in the opposite direction. So when there is equilibrium established between this chemical force and the electrical force, this is called as This equilibrium is called as Nernst potential. This equilibrium is called as Nernst potential. This equilibrium is called as Nernst potential. So Nernst potential is, सो नरेंस पोटेंशियल जो है ये एक इम्पॉर्टेंट कंसेप्ट है जो हमें पता होना चाहिए कि क्या है कि नरेंस पोटेंशियल इज द पोटेंशियल एट विच देयर इज नो actual movement, or there is no net movement of the ions taking place across the membrane, because some of the ions are moving due to the concentration gradient while in one direction, and some of the ions are moving due to the electromotive force in the opposite direction, so net movement is not taking place. So Nernst potential is the diffusion potential that exactly stops net diffusion of an ion, and the concentration gradient equals the electrical gradient. And this Nernst potential is calculated by the Nernst equation. And the Nernst equation is that Nernst equation is plus minus 61 log into concentration inside divided by concentration outside. So in case of potassium ion, normally there are more than one ions which are operating along the membrane in the living cell. However, when we are calculating the Nernst potential for one ion, we are considering a hypothetical condition in which only one ion is present, while there is no other ion which is moving along the membrane. So when cell membrane is only permeable to potassium ion and not to any other ion, this is not possible in a living cell, only an experimental condition. तो इसमें क्या होगा कि नरेंस पोटेंशियल अगर कैलकुलेट की जाए तो कंसंट्रेशन इनसाइड जो है वो 140 मिली इक्विवेलेंट होती है और कंसंट्रेशन आउटसाइड 4 मिली इक्विवेलेंट होती है. तो जो नरेंस पोटेंशियल आएगी it comes to be about minus 94 millivolt. So Nernst potential due to the movement of only potassium ion is equal to minus 94 millivolt. इसी तरह नरेंस पोटेंशियल फॉर सोडियम आयन अगर कैलकुलेट की जाए, सिमिलरली इट इज़ अ हाइपोथेटिकल कंडीशन. ठीक है, क्योंकि देर आर अदर आयंस विच आर आल्सो ऑपरेटिंग अलोंग द सेल मेम्ब्रेन. तो इसके अंदर जो नरेंस पोटेंशियल आएगी, नॉर्मली द कंसंट्रेशन ऑफ सोडियम आयन इनसाइड द सेल इज 14 मिली इक्विवेलेंट पर लीटर एंड कंसंट्रेशन आउटसाइड इज 142 मिली इक्विवेलेंट पर लीटर. तो जो नरेंस पोटेंशियल आती है ड्यू टू द सोडियम आयन, इट इज अबाउट प्लस 61 मिली वोल्ट. तो Nernst potential for sodium ion is plus 61 millivolt and Nernst potential for potassium ion is minus 94 millivolt. And the effect, so here we we can see that in the living cells they are both permeable to sodium, potassium, and chloride as well, and their combined effect determines the net potential difference. For that, we use another equation which is called as Goldman-Hodgkin-Katz equation, in which the resting membrane potential comes to be about minus 61 minus 86 millivolt due to the effect of sodium, potassium, and chloride ion. The net potential that is derived from the Goldman-Hodgkin-Katz equation, the electric motive force comes to be about minus 86 millivolt. अब इसमें हम देख सकते हैं कि जो सोडियम का इफेक्ट है, इट इज़ मच मोर एज कंपेयर टू द इफेक्ट ऑफ अदर आयन, बिकॉज सोडियम आयंस आर the sodium channels are much more predominant and sodium ions are much more permeable through the sodium channels. So value in large myelinated nerve fiber, जो सोडियम पोटेशियम पंप है, उसका रोल है -4 मिली वोल्ट का, और कंबाइंड पोटेशियम और सोडियम का जो इफेक्ट है, इट इज़ अबाउट -86 मिलीवोल्ट, एंड द टोटल नेट वैल्यू ऑफ द रेस्टिंग मेम्ब्रेन पोटेंशियल इज -90 millivolt. Potassium contributes more than sodium in establishing the resting membrane potential due to high permeability. Resting membrane potential, all the three processes, sodium potassium pump, potassium diffusion, and sodium diffusion, keep working simultaneously and lead to steady state condition at resting membrane potential of about -90 millivolt. Sodium and potassium ions have active pumps and have large concentration gradient and large permeability. Due to lack of these characteristics, contribution of ions other than sodium and potassium is negligible. और अगर पोटेशियम कंसंट्रेशन इंक्रीज कर जाए, इफ देर इज हाइपरकेलेमिया, तो रेस्टिंग मेम्ब्रेन पोटेंशियल विल बिकम लेस नेगेटिव. सिमिलरली, इफ देर इज हाइपोकेलेमिया, अगर पोटेशियम की कंसंट्रेशन एक्स्ट्रा सेलुलर फ्लूइड में कम हो जाए, तो इट विल लीड टू रेस्टिंग मेम्ब्रेन पोटेंशियल विल बिकम more negative. The resting membrane hypokalemia में resting membrane potential will become more negative, and in the hyperkalemia resting membrane potential will become less negative. सो थैंक यू वेरी मच. ये कुछ हमारा रेस्टिंग मेम्ब्रेन पोटेंशियल के ऊपर बात हुई. इसके बाद हम इंशाल्लाह ग्रेडेड पोटेंशियल और फिर एक्शन पोटेंशियल के ऊपर बात करेंगे नेक्स्ट लेक्चर में. थैंक यू सो मच फॉर लिसनिंग.